Hello and welcome to this section on our tutorial on collecting and reporting microscope metadata with Micro-Meta App and MethodsJ2. My name is Joel Wright, and in this section, we will discuss a Fiji script called MethodsJ2, a software tool to capture metadata and generate comprehensive microscopy methods text. MethodsJ2 runs in Fiji and uses hardware metadata generated in Micro-Meta App, which was described in a previous video by Caterina Strambio de Castilla. I'll give you an outline for this series of videos. First, we'll discuss the importance of methods reporting. Then we'll give an overview of MethodsJ2 and its data inputs, including the Micro-Meta App hardware specifications file. Then we'll go over the materials needed for this tutorial and the things to download. And finally, we'll walk through MethodsJ2 with demo files, so an actual hands-on tutorial. One problem that we wished to address is that method sections for microscopy experiments tend to be incomplete. This problem was known to the imaging community and more thoroughly investigated by some of our colleagues in their 2020 paper, Imaging Methods Are Vastly Underreported in Biomedical Research. One of their main findings is that out of about 240 papers examined, less than 20% of those papers contain enough information to replicate microscopy experiments. Of course, this is not ideal. Complete method sections, including information about sample preparation, microscope hardware, and acquisition settings, are important. First, it helps evaluate the work and the conclusions drawn from imaging experiments. Secondly, it enables reproducibility, allowing others in your lab or elsewhere to replicate and build upon those findings. Thirdly, complete method sections and well-curated data sets promote data reusability, something which is a bit more common in the world of next-generation sequencing compared to imaging. And finally, slowly but surely, publishers are starting to require that method sections be more complete and contain enough information for reproducibility. So for a novice user, it can be daunting as to where to start. So if you're just starting out with imaging, with microscopy, in terms of reporting method sections, we recommend the following papers. So first, as I mentioned earlier, imaging methods are vastly underreported in biomedical research. This includes a tool to help generate a method section based on image metadata. And we also recommend, of course, best practices and tools for reporting reproducible fluorescence microscopy methods, which is a bit more of a guide of what hardware and what information to include, along with another tool to help generate a checklist of important parameters to include. Building on these findings, recommendations, and tools, and using Micro-Meta App files, we'll discuss in this series MethodsJ2 and how it can help with writing method sections. This ends the introduction. I'd like to acknowledge everyone who has worked on MethodsJ2 and Micro-Meta App, and in particular Claire Brown and Caterina Strambio de Castilla for their guidance and vision. In the next video, we'll give an overview of MethodsJ2 and discuss data sources.